guys. Today we are going to talk about CDK4/6 inhibition in cancer and beyond, and how it helps in cell cycle arrest. So we are going to talk about whether a CDK inhibitor is going to kill all cancer cell types or it is dependent on different cell types, right? So basically, what happens is CDKs are involved in cell cycle regulation, and if you inhibit these proteins, CDK proteins, the cell cycle arrest happens and the cell gets killed. If you just look at this mechanism, it is obvious that it should kill all the cancer cells, but we know that cells are made up of different proteins and protein interaction networks, which results in very complex mechanisms, and due to which it is already known that the CDK inhibitors are basically sensitive against tumor cell types, right? So, if we talk about a specific CDK inhibitor such as abemaciclib, right? So, this paper shows that this is it is resistant against some cell lines. For example, these cell lines, it is very much resistant, and for HT29, CACO2, and SNUC14, C4, it is very much sensitive, right? So, and this is very well known fact that that this inhibitor is sensitive against some cell lines and resistant against other cell lines. So, the question which gets posed is, uh, why we are talking about this. We are talking about this because suppose you are a pharmaceutical company, and you have a new novel drug against a CDK4, and you want to test in which cell lines it is going to work, right? So whether it will work in some cell lines or all cell lines, or and if it works in some cell lines, which of these are cell lines, right? So a very simple use of our software data discover, which shows this is the result, and how we got the result you can see in the previous videos. You just have to input the simple smile of this, the smiles which we have taken from this pubchem, right? And then these are the IC50 values in nanomolar against different 75 different cell lines. We can see that it is sensitive. IC50 values are less in some cell lines and more in some cell lines. So in cell lines it is more, that means it is drug resistant, and in less, that means it is, for those cell lines it is sensitive. So for example, this relay cell line which is a 5000 nanomolar IC50, it is going to be very much resistant, right? So if I download the text file and show you the result in an Excel sheet sorted, so we can see that for all these different cell lines, so this is a protein, so it doesn't matter. So all these different cell lines, I've sorted the results based on the IC. These are the IC50 values in micromolar. So that means for all these cell lines, it is going to be very much sensitive. And if we go down, for all these cell lines, it is going to be very much resistant. This is same as these results. For example, HCT15, it is going to be resistant, and HT sensitive, right? So if you look at this, so this is my HT29 and this is my HCT15, right? So HT29 is above and HCT15 is below. That means the results are not exactly quantitative because it's an AI model. So, but a ranking of the cell line shows gives you an idea of which cell line you should work first, right? So this is a sensitive cell line, and these are the resistant cell lines, right, which is same as the experimental result. So basically this gives a pharmaceutical company an idea of, instead of doing all 75, purchasing 75 cell lines and doing experiments, you can just focus on a subset of cell lines and do your further experiments, right? So with this we end. Thank you.